This video covers the topics of DNA structure and replication. So just as I stated, we will go over the structure of DNA and then how a DNA double helix is replicated to make two new DNA double helices. So you will need to be able to identify the parts of a nucleotide, describe the three differences between DNA and RNA. You need to be able to describe the structure of DNA, the concept of complementary base pairing, and how this applies to DNA replication. And then finally, three different enzymes that are involved in the process of DNA replication. And those would be DNA helicase, DNA polymerase, and primase. So you were introduced to the parts of a nucleotide during the biological molecules lecture. So here is the same image that I used during that presentation. So the nucleotide is the building block of a nucleic acid. Now, a nucleotide is composed of three different molecules. So you have a phosphate group, a sugar, and a base. Now they call it a nitrogenous base, which basically just means it has a nitrogen in it. So here you can see that. I just refer to it as a base. Now you need to know which bases are present in DNA. And then we'll also talk about the bases present in RNA. But let's start with DNA. So there are four different bases in DNA. And like I said, you were introduced to these during the lecture on biological molecules. So I want you to take a second and think about what these four different bases are. Now you can see the A, G, C, or T from the image that I provided. So A is for adenine, G is guanine, C is cytosine, T is thymine. So what this means, there we go. What this means is that in a single nucleotide of DNA, you're going to have that phosphate group, the sugar, and for DNA, that sugar is called deoxyribose, and one of those four bases, adenine, guanine, cytosine, or thymine. Now, you were also introduced to this information during the biological molecule lecture, but you need to know what the three differences are between DNA and RNA. So take a second here and look at the picture of DNA and compare it to the one of RNA and see if you can remember what those three differences are. Okay, so the first difference between DNA and RNA is that DNA is double-stranded. It is what we call a double helix, but RNA is single-stranded. So I'm going to write DNA double-stranded, RNA single-stranded. So basically what that means is that DNA will have a strand of nucleotides, and then that strand of nucleotides is bonded to another strand of nucleotides. But RNA does not have that structure. Now, another difference between DNA and RNA is with the sugar. So DNA has deoxyribose as the sugar. RNA has ribose. That's why DNA is deoxyribonucleic acid, RNA is ribonucleic acid. So that reflects the fact that they have different sugars. Now, these sugars are almost entirely the same, except the deoxyribose in DNA is missing one of the oxygens that ribose has. Now, the last difference, or the third difference between DNA and RNA, and this is really important that you remember this, is that DNA has thymine as one of its bases, and RNA has uracil. So thymine is not present in RNA. Instead, it is uracil. Now, this is really important when we talk about protein synthesis, where an RNA is created from a DNA template. So just to summarize, there are three differences between DNA and RNA. DNA is double-stranded, RNA is single-stranded. DNA has deoxyribose as its sugar, RNA has ribose. And then finally, DNA has thymine as a base, RNA has uracil. Now, remember that DNA and RNA still have adenine, guanine, cytosine. Those three bases are similar between the two. But DNA has thymine as its fourth base, RNA has uracil. So now we are going to look at the actual structure of DNA. So what you are looking at here on the left is a DNA double helix. So you're gonna have to bear with me with this drawing. I have a Mac now instead of a PC. It doesn't have a touch screen, so I can't really draw on my slides. So I'm going to use like little arrows and things to show you what I'm talking about. So here we have a DNA double helix, like I mentioned. So if you look at the picture here, it says sugar phosphate backbone, and it has like a yellowish color, it has those going down the side, and in the middle, it says base pairs. Now, what I want you to notice here is that there is a rectangle in the lower left-hand corner that says nucleotide. So what you're looking at here when you look at one side of the double helix is a nucleotide attached to a nucleotide attached to a nucleotide. So let's take a look here. So what I'm showing you here, where I drew this circle, this would be considered one strand of DNA. Okay, so we have the nucleotide on the bottom, phosphate, which is symbolized by a P, sugar, symbolized by an S, and base. It shows a C, meaning that base is cytosine. Now, the phosphate on that nucleotide covalently bonds with the sugar of the next nucleotide. So let's take a look here. So here is your next nucleotide where I drew this oval here. You have a phosphate, a sugar, and the base is thymine. Now the next nucleotide in that strand is right here. So again, the phosphate of the previous nucleotide is covalently bonding to the sugar on the next nucleotide. So you have a phosphate, a sugar, and then the base is guanine. So hopefully you're getting the idea that this is one strand of DNA, but nucleotides are the building blocks. It's nucleotide bonded to a nucleotide, bonded to a nucleotide. So let me erase my circles here. Now, I told you that DNA is double-stranded. So here, I'm going to draw a circle here. This, where I just drew my oval, is the other strand of DNA. Now, one strand of DNA is attached to the other strand of DNA in the middle by forming hydrogen bonds between bases. So you can think of it like this. The DNA double helix is kind of like a ladder, right? The sugar phosphate backbone, where it goes phosphate, sugar, those are like the sides of the ladder that you would hold on to. The bases project to the inside of the double helix, and they are hydrogen bonded together. So that would be like the rungs of the ladder, like the steps, right, that your feet go on. So the part where you put your hand on the ladder, and then the rung is like a one strand. And then you have the other part of the ladder. The part where you put your foot on is the other part with the bases, and they're attached in the middle. Hopefully that makes sense. So now let's look at how these bases attach to one another. So in the middle, the bases attach to each other through complementary base pairing. So I'm going to write down some information here for you. So one strand of DNA is attached to the other strand. Actually, I'm going to write one strand of DNA attaches to the other strand. Using hydrogen bonds between the bases. Now, this follows complementary base pairing rules. So let me show you that. So we have complementary let's see, I need a little more room for this. So complementary base pairing rules. That means that if you have an adenine as your base on one strand, it always hydrogen bonds with a thymine on the other strand. So we're going to say adenine always pairs with thymine. Okay. Cytosine always pairs with guanine. Oh, hopefully you can see that. That's going into some of the other colors there. So A always pairs with T, C always pairs with G. So I'm going to go to the whiteboard to illustrate this. Oh, wait a second here. One second here. You have to do it in a very specific way in order to get it to work. And that did not work. There we go. You should be able to see my whiteboard now. Okay, so let's draw a strand of DNA across the top of my whiteboard and a strand of DNA across the bottom. Now I'm representing it with just a line, but remember it goes phosphate, sugar, base, phosphate, sugar, base, right? Nucleotide bonded to nucleotide, bonded to nucleotide, okay? So now I'm going to draw these short little lines to show you the bases that are projecting into the middle of the DNA strand. I drew myself a very long line here. Okay, I'll put one more base. And we're gonna do this. So bear with me here while I get this drawn out. And actually, when I draw things on the whiteboard, I think it's a really good idea for you to draw them in your notes. I mean, you do always have these by going back to my recordings, but the actual practice of writing them down will help you understand and remember them. Okay, so now I'm going to add some bases. We've got G, let's do an A. Oops. How about another A? We'll do a C. So I'm just making this up as I go, right? It can be any sequence. So I want to put this in kind of a bigger context for you. Now, remember that when you are looking at DNA, first of all the DNA in your cell is called your genome. And every one of your cells has the exact same DNA, right? It's just how that DNA is controlled that helps your cells differentiate into, say, a stomach cell or a liver cell or a skin cell. But all of your cells have the same DNA. Now, about 5% of your DNA are actual genes. Okay, so genes code for proteins. Now, most of your DNA does other stuff, like control those genes, meaning that sometimes the genes are working, sometimes they're not. Sometimes they're producing proteins, sometimes they're not, right? But only 5% of your DNA actually codes for proteins or are considered genes. Now, a gene can be thousands of nucleotides long, could be hundreds of nucleotides long. It just depends on the gene, okay? So here we have our top strand of DNA. It has the base guanine, adenine, adenine, cytosine, guanine, thymine, thymine, cytosine. Now remember that I told you that these DNA strands connect to each other by hydrogen bonds between the bases, but they have to follow base pairing rules. So I'm going to write these base pairing rules down here for you, even though I had it on the other slide. So this is what we call complementary base pairing. By the way, the word complementary doesn't mean like compliments, like, hey, you look really good today. It means complementary, like they go with each other, right? They complement each other. So complementary base pairing. A pairs with T, C pairs with G, always. So we have a G on the top strand, which means that you have to have a C on the bottom, right? Guanine always pairs with cytosine. If you have an A on the top, you're going to have a T on the bottom. So here we have an A, which goes with a T. I'm going to move this T so it's a little easier to tow. There we go. Okay, we have a cytosine on the top, which goes with a guanine on the bottom. Guanine pairs with cytosine. You should be getting the hang of this now. Thymine pairs with adenine. Here we have another thymine, so it pairs with another adenine. And then finally, a cytosine that pairs with a guanine. Now, I do want you to remember that when I say pair and bond, I'm talking about hydrogen bonds. So the guanine, now remember hydrogen bonds, we symbolize those with a dotted line, and I'm not going to draw dotted lines on all of these because it takes a really long time. Okay, but these bases are connecting to each other with hydrogen bonds. Now, when you look at that sugar phosphate backbone for one strand of your DNA, those are covalent bonds. They're strong, but hydrogen bonds, those are weak. And we'll talk about why that is important when we get to the actual DNA replication. So this is all a precursor to helping you understand DNA replication. So let's go back to the screen on the PowerPoint. Hold on one second here. There, share. Huh. Okay, you should be able to see my PowerPoint screen now. I had a few technical issues there. So once again, you are looking at a DNA double helix. So if we go back and look at this picture here, you can see that here we have a guanine and it is paired with a cytosine. Here we have an adenine paired with a thymine. Okay, so complementary base pairing. And notice the dotted lines in between the bases, those are hydrogen bonds. So that is important for you to remember. Okay, so far we've looked at DNA structure. And for DNA structure, whoops, go back. DNA structure, you need to know that the nucleotide is the building block of DNA. It has a sugar phosphate base. Okay, so for DNA, you have a deoxyribose sugar, the phosphate group, and then the bases are adenine, guanine, cytosine, thymine. Now, I also showed you the three differences between DNA and RNA. DNA is double-stranded, RNA is single-stranded. DNA has deoxyribose as its sugar, RNA has ribose. And then finally, DNA has the base thymine, but RNA has the base uracil. So now let's go on into DNA replication. Now this can also be called duplication or synthesis because during this process, the amount of DNA is being doubled. Now, big picture here, DNA replication occurs prior to cell division or cell reproduction, right? That's how you have like a skin cell and it can make more skin cells. Okay, so DNA replication occurs before the process of cell division. Now, what I'm showing you here in this picture is one DNA strand will act as the template for a daughter DNA strand. So here we have in the picture a DNA double helix, right? That's how your DNA normally looks. Now, during replication, that DNA double helix will separate. So these bluish strands are what we are going to call the parent or old strand. Okay, so here's the blue strand. This is from the original double helix. Now, from that original double helix, a daughter DNA strand is produced. So this old strand on top is used to make a daughter strand. The old strand or template strand on the bottom is used to make another daughter strand. So you start with one double helix, you end up with two double helices, that's plural, and they are exactly the same as the original double helix. That is pretty much the entire function of DNA replication. Let's go on and look at this. So this is an image I found that I think really summarizes DNA replication pretty well. So you have what it says here on the left, the DNA double helix is separated. Did I spell that right? I think I did. So it's separated by breaking the hydrogen bonds between the bases. So let me repeat that for you. The DNA double helix is separated by breaking the hydrogen bonds between the bases. So I'm actually going to write that for you here because it is important that you remember that. So the DNA double helix is separated by breaking the hydrogen bonds between the bases, okay? Between like the adenine and thymine, cytosine and guanine. Now, each DNA strand from the original double helix is used to make a new DNA strand. So those original strands are called the templates. The new strands are the daughter strands. So each new double helix contains one old strand, one new strand. Now, if you look this up online, it will also be called a parent strand and daughter strand. So now I am going to go to the whiteboard for a simplified image of how this works, or drawing, I guess I should say. So let's go here. All right, we'll go to the next page. Okay, I'm gonna try to draw this with lines and stuff. It's not gonna be quite as easy as if I could draw on my screen. But let's say this is our original DNA double helix right here. Okay, I'm gonna draw some bases on here. Actually, I'm not gonna, let me not draw the lines here. I'm just gonna write the bases. So in the interest of time. All right, so we have an A, which means we'll have a T down here. Remember, complementary base pairing. Let's do a C, which means a G here. Oops. Okay. T, T, almost done writing my bases here. And let's do a G as our last one. Okay, so now we're going to complementary base pair those. So you should know that a T goes with an A. The next thymine goes with an adenine. T always base pairs with A. And guanine always base pairs with cytosine. Okay, so this is our original DNA strand. So we're looking at replication. I'm going to draw this kind of like that. So the hydrogen bonds between the bases, they broke. And those DNA template strands separated so that daughter strands can be produced. I'm going to put some bases on here. A. This is going to be a little messy because it's taking me too long to move everything around. Okay, those are the bases on that template strand, A, T, C, G. Now on this lower template strand, remember, it's still base paired, right? Even though you broke those hydrogen bonds, the bases still follow those base pairing rules. So it would be a thymine, an adenine, guanine, and finally cytosine. Okay, so let me label these for you. So down here we have a template or old strand of DNA. And up here is a template or old strand. Okay, now through DNA replication, 2 new daughter strands will be produced. And then those will form the 2 new double helices. So let's look at how that works, because these 2 new double helices are exactly the same as the original DNA double helix. So I'm going to draw this. Oh, maybe I should have made this bigger. It's going to be kind of hard to fit it in. Okay. So here we have adenine on the template strand on top, which will base pair with thymine, right? Always base pairs with thymine. Another adenine, so another thymine on the daughter strand. Thymine, so you have adenine on the daughter strand. Then comes guanine. And finally, cytosine. So what you should notice here is that the new daughter strand actually has the same nucleotide sequence as the other template strand. So let me label these, and then I'm going to draw some arrows to show you what I mean. Okay, so where I just wrote daughter DNA strand, that looks exactly the same as this template strand right there, okay? Because these are exactly the same double helices, right? You're just doubling the amount. So now I'm gonna draw the other DNA daughter strand, but I wanna move this around a little bit because I didn't give myself enough room. So let's see, that was down here. Okay, now I'm gonna move this, whoops. For some reason, it's not letting me move my, oh wait, here we go, okay. Just trying to give myself a little more room to write the nucleotide sequence. Okay, So now I'm looking at the bottom template strand. So you are getting a daughter strand, a new daughter strand based on that template strand. So for a T, you get an A. The next T base pairs with an A. T. Okay. So what you should notice here is that the two new double helices look exactly the same, right? Okay, you have A, T, C, G on the top, T, T, A, G, C on the bottom. Now, for those two new double helices, there are hydrogen bonds between the complementary base pairs. So you went from one double helix to two. That is the entire purpose of DNA replication. So let's go back to the PowerPoint. Okay. I put an Amoeba Sister video in here, and like I always tell you, these are really great because they are super straightforward. The other thing I like about these videos is they give you mnemonics to try to remember the things, like the different enzymes, stuff like that. So like if you look at this picture here, it has the enzyme ligase. It calls it the gluer because it, puts little DNA segments together. So I like this video because it gives you ways to remember the information. Now, keep in mind that this video goes into more detail than I'm going to go into, so focus on the parts that are actually in this PowerPoint. Now, I mentioned that you need to know 3 different enzymes for DNA replication. So it's actually really a complicated process. I'm only asking you to know very few things about the actual process of DNA replication because you really don't need to know it super in depth. So here are the 3 enzymes I would like you to know. The first enzyme is called DNA helicase. Now remember, enzymes end in ASE. So DNA helicase, according to the Amoeba Sister video, is called the unzipper. What that means is that this enzyme breaks the hydrogen bonds between bases of the original double helix. This is how the double helix opens and the 2 new daughter strands can be formed. So let me summarize that for you. The unzipper breaks the hydrogen bonds. I know you can't see this, so, oh yeah, you can. Thought I was gonna have to move it. Breaks the hydrogen bonds between bases in the original double helix so that the daughter strands can be produced. So DNA helicase, it breaks those hydrogen bonds so that the 2 DNA strands in the original double helix can separate and the daughter strands can be produced. Now, the next enzyme I would like you to know about is called primase. Actually, I'm going to jump ahead to DNA polymerase, and then we'll go back to primase. So DNA polymerase is on the bottom. The function of DNA polymerase is to add nucleotides to the growing strand of DNA. And by growing strand, I mean the daughter strand. So let me write that down. Then I will talk about it a little bit more. Now put in parentheses, daughter strand. So just like I tried to show you on the whiteboard, when the two DNA strands separate, right, they are now template strands, nucleotides come down from, this is occurring in the nucleus, so there's kind of like nucleotides just hanging out in the nucleus. So DNA polymerase brings the appropriate nucleotide, the one that base pairs with the template strand, and adds it to the daughter strand so that it grows. Okay. So in the picture here, it's showing you the two template strands of DNA. They're separated, right? Where it says free nucleotides, those are just like phosphate sugar base, right? That's a nucleotide. Now, DNA polymerase will add the correct nucleotide to the new strand. And by correct nucleotide, I mean the one that base pairs with the template strand. So if you have a thymine on the template strand, DNA polymerase will add a nucleotide with an adenine to the daughter strand. Now, to see this, like to visualize it, watch that Amoeba Sister video. The last enzyme we are going to talk about is primase. See, I'm going to talk about that right here. So primase adds a really short segment of nucleotides that shows DNA polymerase where to start the daughter strand. Now, the tricky thing here is primase adds RNA nucleotides, not DNA. And what that means is that this really short segment of nucleotides has phosphate, sugar, base, but the sugar is ribose, and the base can be adenine, guanine, cytosine, uracil. Now, this short little segment of RNA primer shows DNA polymerase. You know, here's where you attach and start that daughter strand. So let me summarize that for you. So primase adds a short segment of RNA nucleotides to show DNA polymerase where to start producing the daughter strand. I'm going to stretch this out so you can see it here. Okay. There we go. So primase adds a short segment of RNA nucleotides to show DNA polymerase where to start producing the daughter strand of DNA. Okay, you do not need to know why it's an RNA primer rather than a DNA primer. That is way too detailed for what you need. So just to review, you need to know for this module, the structure of DNA, the three parts that are in a DNA nucleotide: phosphate, sugar, base. Okay, there are four bases that go into DNA: adenine, thymine, cytosine, guanine. The sugar is deoxyribose. You need to know the three differences between DNA and RNA. DNA is double-stranded, RNA is single-stranded. DNA has deoxyribose for its sugar, RNA has ribose. And then finally, DNA has thymine as one of its bases. RNA does not have thymine, it has uracil. Now, with regards to DNA replication and also the structure of the DNA double helix, you need to understand complementary base pairing. Adenine always pairs with thymine, cytosine always pairs with guanine. They are held together by hydrogen bonds. Now, hydrogen bonds are weak, and that is really important for DNA replication because it is easy for the enzyme DNA helicase to break those hydrogen bonds. You want them to be weak. Covalent bonds are strong, hydrogen weak. Okay, you need to know that the DNA double helix separates. You have two template strands. Daughter strands are built off of the template strand, and you end up with two new DNA double helices that are exactly the same. And they're the same as the original DNA double helix. This is doubling the amount of DNA in the nucleus of your cells prior to cell division. And then finally, you need to remember what these three enzymes do: DNA helicase, primase, DNA polymerase. Okay, my dogs say we are done. Let me know if you have any questions.